let's have a look, in the same way as we looked at bacteria, which bits of the fungal cell or the fungal metabolism are sufficiently different from our own cells that they would make a useful target for antifungal drugs. Here's a generalized fungal cell. As you can see from the first picture that I showed you, there's a huge diversity within the fungal kingdom, but generally their cells look like this. They have a cell membrane, that's the pink thing there, which is similar to our cell membranes in that it's a lipid bilayer, but there is a difference between the composition of their lipid bilayer and our lipid bilayer, and that is that in the mammalian cell membrane, cholesterol is one of the major lipids that forms that lipid bilayer. In fungi, it's a slightly different sterol, it's ergosterol. Rather than cholesterol, one of the major lipids in the cell membrane is ergosterol. So there straight away is a target. If you could target drugs towards the ergosterol component of the lipid bilayer in fungi, then you would have a drug that would not damage our cell membranes, because we don't have ergosterol in our cell membranes. And in fact there are two groups of antifungals, the polyenes and the azoles, which act in this way. They act by disrupting the cell membrane by targeting ergosterol. And those are probably the leading antifungals that we have in terms of use. Fungi, of course, synthesize DNA and RNA, as do any cells which are going to have to reproduce. And there is an antifungal drug which acts to block DNA and RNA synthesis in fungi, that is flucytosine. We'll talk about why that is able to target specifically fungal DNA synthesis as opposed to anybody else's DNA synthesis when we get to that point. So that's another group of antifungals. And finally, fungi have a cell wall in the same way as bacteria have a cell wall. There's a number of differences between the fungal cell wall and bacterial cell wall and plant cell walls, in that the cell wall actually contains chitin, which is a protein which makes up the hard exoskeletons of insects. You know, that hard, brittle outer skeleton that insects have. And the fungal cell wall contains chitin rather than cellulose, which you find in plant cell walls. But anyway, the cell wall is also obviously a potential target, because we don't have cell walls at all. And there are antifungal drugs which target cell wall. Caspofungin is an example. But in terms of the use, it's the ones that target the cell membrane of the fungus, the polyenes and the azoles, which have the most use as antifungals at the moment. So let's have a look at those, the ones which act by disrupting the fungal cell membrane. And the first group I'm going to deal with are the azoles, because they are the most widely used at the moment. They're called the azoles as a group, and look what they've got in their generic names. Very, very handy. Fluconazole, clotrimazole as I call it, ketoconazole, voriconazole. All the onazoles are the azole antifungals, and they act by attacking the fungal cell membrane. They do that by inhibiting the synthesis of ergosterol, that is the sterol, the fatty substance that the major part of their cell membrane, and therefore the cell membrane integrity is disrupted. The cell membrane starts to pull apart. So the azoles are very good for that. They're relatively safe to use, relatively few adverse effects in us, because we don't have ergosterol in our cell membranes, which is very handy. So you can use them systemically. There are both systemic and orally available azoles and topically available azoles, so you can use them to treat both systemic infections and topical infections. They are broad spectrum. They are active against a lot of different fungi, which is also very handy, and they have a wide therapeutic index. That's excellent. All of those things are good. Another good thing about them is that they're not just confined, some of them, to antifungal effects. They can have other effects against protozoans and some bacteria as well. So they're even broader spectrum than the fungal kingdom. So you can see by looking at just a few of the headlines about the azole group why they would be the most commonly used antifungals. Here's some pictures of available products which contain azoles. You can see the ketoconazole tablets there. This is a shampoo containing ketoconazole. Here's one for treating vaginal thrush and other fungal infections around and about the place, oral thrush and dermatomycosis, infections on the surface of the skin, and that's fluconazole. This is one which is very well known, canesten, the clotrimazole, antifungal cream, and then we've got this one voriconazole that is available by injection only. So all sorts of azoles to treat all sorts of fungal infections. So, okay, the next bunch that also act by disrupting the fungal cell membrane are the polyenes, and notice they don't have the helpful enes ending. There are pretty much only two that I use, amphotericin B and nystatin, and they are again targeting ergosterol. They don't inhibit the synthesis of ergosterol, but they bind to ergosterol, and by their binding they kind of mess up the membrane because they form a sort of big lump in the membrane and make it more permeable. They actually kill the fungi at high doses, and they're fungistatic in lower doses. The problem with the polyenes is that they are pretty toxic, so you're not going to use them systemically. You're not going to introduce them into your bloodstream unless you really have no choice. In other words, these would be kept for systemic use only if there was a life-threatening invasive fungal infection, or these would be your fallback antifungals for systemic infections. Nystatin is available for topical use. The toxicity, if you're using it topically, is not going to be such a problem, or for gastrointestinal fungal infections. Now, when it's used for that purpose, it isn't actually absorbed. Nystatin isn't absorbed, so it remains in the gastrointestinal tract and treats any fungal infections that are there. There shouldn't be any systemic absorption if you're using it topically or for gastrointestinal mycosis. And you might be thinking, oh well, if they're binding to ergosterol and we don't have it, then why are they so toxic? Well, it's one of the constant surprises with drugs. Sometimes you think a drug is targeting a particular type of substance that you don't have, and then you find it's actually targeting something else as well, and it produces more effects than you wanted it to. So those are the polyenes. I'll just show you some of the products available. You can see the nystatin cream, topical use, so that's okay. Nystatin oral drops, vanilla flavoured. These would be used to treat oral thrush, for example, but again not to be absorbed. And here's the amphotericin B for injection, and you wouldn't go there unless you really had to. So those two groups of drugs are the antifungals that operate by disrupting the fungal cell membrane. Now if we move to antifungals which operate by disrupting the cell walls, that thick chitinous cell wall, these are drugs known as fungins, caspofungin and anidulafungin, again competing well in the world impossible to pronounce drug contest. But they are basically inhibiting the normal synthesis of the fungal cell wall in the same way that we saw that the beta-lactams inhibit the normal synthesis and maintenance of the bacterial cell wall. These are doing an equivalent thing in the fungal cell wall. Again, these are not things that you would use as your first choice. They are only available for intravenous administration. There are no oral versions of them, and you would only use them if you had strains of aspergillosis or candida that were invasive and that were resistant to the other antifungals that we've talked about. So that's the fungal cell wall. And then finally, the drugs that act by disrupting fungal DNA or RNA synthesis. And it's just one. It's flucytosine. Now I'm mentioning this because obviously it is an antifungal, but also because we're about to do the cancer chemotherapy. This is a little bit of a taster of an anti-cancer drug, because flucytosine is actually a drug that is known as a prodrug. In other words, it's not in its active form when it's given, so flucytosine is not actually the active drug. It needs to be converted once it's been absorbed to the active drug by the action of some enzyme that exists in the fungus. And this is where the clever part comes in, because it's actually converted by the fungus, once it gets into the fungal cell, to the active form, which is 5-fluorouracil. Now 5-fluorouracil is cytotoxic. In other words, it will kill cells. It would kill our cells. So if you gave the drug as 5-fluorouracil, you would be killing lots of healthy cells of your own. But because you can give it as this prodrug, and only the fungal cells possess the enzyme to convert it from flucytosine to 5-fluorouracil, it does actually target to the fungal cells themselves, because none of your cells have the capacity to convert flucytosine to the cytotoxic 5-fluorouracil. So it's quite clever. It gets itself incorporated into RNA as RNA is being produced, and it results in a defective RNA, which prevents successful replication and division of the fungal cells. It isn't widely used against fungi, mainly because it doesn't have a broad spectrum of activity, so it's only active against certain fungal infections anyway, plus resistance is starting to come out, which limits its use. So if you see it used, it's usually used in combination with another antifungal to try and increase the overall antifungal effectiveness. So resistance, we haven't talked about resistance in relation to fungal organisms, but just like bacteria, of course, if you keep nobbling a certain fungus or certain range of fungi with a certain drug, then you are putting selection pressure on those organisms to produce resistant strains. The cells that contain some kind of genetic advantage that helps them to survive the effects of that drug better are going to be the ones that survive. So with continued use of a particular antifungal, you are going to breed a resistant strain. And so that is happening just as it is with antibacterials in the bacterial world, it is also happening in the fungal world. Just to bring that point home, in the same way as bacteria acquire resistance, fungal cells are really no different. So here's an example of a fungal cell being acted on by those azoles. Now normally you'd expect them to get in the way of a normal ergosterol synthesis and block it. And in a fungal cell normally there would be pumps, active transport mechanisms in the membrane of the fungal cell which could actually remove some of the antifungal drug. But they're at a level at which you can still achieve an effective concentration inside the fungal cell. But with continued use, you start to put selection pressure on, and you lead to an increase, which leads to an increased number of resistant fungal cells, and their resistance comes about in similar ways to the way that bacteria acquire resistance. For example, the enzyme which is involved in ergosterol synthesis to which the drugs bind has a slight conformation change, so that the drugs no longer have that same inhibitory effect on the ergosterol synthesis. And there can be many more of those efflux pumps put into the membrane, so a much greater number of efflux pumps, which means that they can get rid of the drug much faster, and it would be much more difficult to ever achieve an effective concentration of antifungal drug in the fungal cell anymore. So very similar means of acquiring resistance as bacteria have. So developing resistance to the antifungals is no less a problem than it would be with bacteria. It's just that we don't have so many problems with systemic infections of fungal type as we do with the bacterial type. So that is it for fungi, just those major drug classes of the antifungals.